But I think we'll, in the interest of time, we'll try to get started. People will kind of phase in, back in again. You know, I actually think, you know, this is one of those things that we probably should do at the very beginning of the course, at the earliest part, because, you know, as I mentioned during my intro lecture, you know, some of our biggest mistakes that our lab has ever made were issues around experimental design, and you kind of learn the hard way. And I think one of the things that will kind of come across in Olga's talk, I think, is the importance and the sheer rigor that needs to be thought about in how you design an experiment, and if you put, you know, poor issues in the initial design of the experiment, basically everything's lost, and that it could end up being enormous amounts of money and resources and instrument time. And we've all done this. So I think it's something that we should be spending more time and think about more carefully. Now, just to give you a little bit about Olga's background, so Olga is a statistician, a true bona fide statistician. There's a lot of us that kind of try to, you know, wing it at times, but Olga is a true bona fide statistician, which I think card carrying.
That's right.
But at the same time, she also did a postdoc in the Aebersold lab, so she's been around mass spectrometry data for most of her independent career. In fact, all of your independent career. She started her faculty career at Purdue, and she's now at Northeastern. She's been teaching this stuff in the context of proteomics and quantitative proteomics now for, I guess it's like 7 or maybe more years. I mean, for as long as our lab for sure has been involved with it, you've been at least involved in that time, if not longer. So I think I've always learned a lot every time watching Olga's lectures, and so, in fact, sometimes it takes me 2 or 3 times to actually kind of get everything. So I definitely think people will appreciate this, and feel free to, again, interrupt as always, and I think having a discussion that leads into coffee or other things later, I think is probably good. So anyway.
Absolutely. Thank you so much. Thank you. I'm very excited to be here. So, as Mike said, I'm a statistician by training, and my interest is all things quantitative mass spectrometry. So we're actually going to have 2 parts today, so before lunch and then after lunch as well. And so what I would like to do in the first part is to talk about general statistical theory and methods for design and analysis of experiments. So it will not be very specific, like a little bit specific to mass spectrometry, but it's really general. But then in the afternoon, it will be more research-oriented and more kind of specific to mass spectrometry, where we talk about the methods which we develop, and you will have a chance to have a hands-on at the end of the afternoon on that as well. So probably some of the things I'll talk about here you have seen at least to some extent, but I think it's really important for all of us to be on the same page. And so as a context, so let's talk about quantitative experiments which aim at relative comparison. So it will be slightly different from what Andy talked about this morning, but we will actually converge back to that at the end of the afternoon. So first of all, why do we need to have this conversation, right? Why do we need to talk about statistics? And the point is that variation and uncertainty are really a hallmark of the data and of the experiments that we have to work with. And so there are all kinds of sources of variation. There's technical variation in sample handling, storage, and data processing, as we have just heard. There's also instrumental variation, so even the same type of sample analyzed on multiple days or different time points on mass spectrometry has variation due to ion suppression, competition for ionization, elution time, and so on. There's also signal processing, because different data processing tools, they make different choices for good reasons, right? But the decisions which are made as far as finding the peaks, quantifying the peaks, identifying the peaks, they also affect our conclusions. So so the 3 types, they are important, but they're all technology related. And so as the technology improves and our protocols improve and our ability to work with these samples improves, we can minimize this type of variation. And so what people tell me sometimes is that if our experiments are done well enough, then we don't need statistics anymore. And my point here is that it's not true, and I really would like to convince you otherwise. And the reason for this is that we are studying biological systems. Our main objective is to understand the function, abundance of proteins in living systems. And there's a natural variation of protein abundance between one person and another, one biological organism and another. And there is a day-to-day variation and person-to-person variation. And so even though our experiments can be perfected to the extreme, we would still have to deal with the biological variation, and this is the most important point. And so what we need to do is to distinguish which variation is just a regular variability in the biological system versus which variation is systematic and is induced by the disease or stress or whatever condition is applied. And so our goal, so the really main objective of statistical mindset is to ensure effective and reproducible research. So reproducible research is really the big theme for us. And statisticians can contribute in at least three ways. So the first way is experimental design. So if we design the experiments with the statistical mindset, we can minimize all sorts of problems which can occur later. We will talk about this quite a bit. Then there's also data analysis. And it's not only about, we have big data, we cannot do it by hand in Excel, let's have some statistical methods to speed it up. It's not about that. It's about making things objective so that our conclusions are not dependent on our subjective opinion, so that they can be repeated and reproduced and criticized potentially, right? And of course, statistical tools. This is also important because if you just write something by hand or do something in Excel and then you forgot where you clicked and what happened, right? So this also undermines reproducible research. So developing tools which facilitate all of that is an important component of what statisticians do. And so you can think of statistics as a body of methods for making wise decisions in the face of uncertainty, and that's exactly what we would like to do. And so just to emphasize that, the point that biological variability is the most important, there's a lot of times in science that we've seen that every time there is a new technology which allows us to quantify more things, faster on a large scale with more sensitivity, somehow people forget that they still need to worry about the biological variation. And this was one example of the RNA-seq technologies or next-generation sequencing, where suddenly people could quantify the abundances of transcripts on a much larger scale more sensitively, and then their sample sizes were 1. Right? And so to the point that these statisticians felt compelled to write a Nature Biotechnology paper saying sequencing technology does not eliminate biological variation. So it kind of sounds silly, but it's really something that we need to think about. And it's exactly the same for us, as our workflows become more and more comprehensive, we still need to worry about biological variation. So that's really the context of what we do. And so what I would like to do before the break is this thing. So first, I will try to set the stage, so tell you what happens in my mind when somebody approaches our group for a collaboration in a particular project. So how can we translate the scientific problem or scientific goal into a statistical goal? Then we'll talk about experimental design. I will review very basic methods for statistical analysis, and then I will spend some time talking about the issues which we can deal with. Okay, so the first thing is how we can define statistical goals. So you can very kind of loosely take all the statistical methods out there and partition them into 3 categories, which have to do something with classes. So classes are either samples or proteins which are similar somehow. And so the first statistical goal is class discovery, where we look at the data and we look for patterns of similarity. And so you have certainly seen things like that. So this is a heat map. These rows are analytes, for example, proteins. The columns are samples. The colors are the abundance of analyte in the sample. And you would say, okay, let me cluster the proteins in the space of the samples, because if proteins have similar profiles, maybe they're involved in the same biological process. Or let me cluster samples in the space of proteins, because if subjects with the same disease cluster tightly, maybe this is a disease subtype which we didn't know about. So these are all meaningful things, all principal component analysis, so this will be also very typical things. So here, each of the axes are some linear combinations of proteins, the dots are biological samples. So if the samples from the same condition cluster together, you say, okay, looks like proteins are informative of the condition. The problem with these methods is that you really don't tell the method what is the property of the class you're looking for. You just essentially tell the method, go and find me something which is similar. Well, this method will find you something which is similar. Now, is this something useful to you? Is this something reflects, let's say, the disease as opposed to batch of the data acquisition, right? So you can very well end up with clusters here which have nothing to do with your question of interest. So these type of methods are really good for quality control for sure, right? They are very good to summarize the data, to visualize the data, and so on. But if, for example, we want to understand which proteins are regulated by stress, or if we look for biomarkers of disease, these are not good methods to look for biomarkers. And I have seen many times people look, for example, at the loadings on principal components. They find protein with high loading, and they say we found biomarker. This is completely wrong because these methods do not have anything specific to the disease. So this is important, right? So these methods can only look for patterns in the data, visualize. QC is great. We always use them for QC. So now the second, and this method is called an unsupervised method because we don't really define what we mean by a useful class. So statistical goal number 2 is a supervised method in that now we label our samples with class membership. So let me just talk about health and disease because it's easy, right? So these are healthy subjects, these are disease subjects. So now each sample is labeled, right, with the class membership. And so you can ask questions such as, on average over biological replicates, is the average protein abundance here different from the average protein abundance here? By the way, I will always talk about the log scale. So average on the log scale is the ratio on the original scale. We get back to that, right? But the important thing now is that we look at the consensus measurements in all the healthy subjects versus consensus measurements in all the disease subjects. And you want to know if on average there is a change or not. And so the methods you would use here will be t-test, analysis of variance, things like that. And the reporting kind of metrics would be p-values, maybe posterior probabilities. So you have certainly seen that, right? And so this is very useful because it allows you to see if there is a difference between the classes of your interest. However, again, if your goal is to do biomarker discovery, and for targeted methods, it's often some kind of question related to that. I have seen a lot of people saying, I have a protein with a small p-value, therefore I have a signature of the disease, or therefore I have a biomarker of disease, which is wrong. And again, I would like to emphasize that protein with small p-values are not biomarkers. And the reason is that, so we need to define what we mean by biomarker, right? And biomarkers are supposed to be actionable, right? So a clinician should be able to get the readout from the measurement and make a decision for the person. Let's say the person has the disease or not, right? And so what we do here, we compare the average protein abundances between groups. But the clinician doesn't see an average patient. The clinician sees this particular patient. And so even if on average there is a change, it may or may not be actionable for the individual subject. And so essentially this is a wrong problem statement, a wrong translation of the problem into statistical terms. So this type of methods are useful, for example, for initial screening or for basic biology where you just want to compare the average patterns. It is not useful for biomarker discovery. And so just to kind of go into this a little bit more, so here's one example where we have two groups and the subjects are tightly clustered around the mean. So if you have a person with this particular protein abundance, it's pretty clear that this person comes from the disease group. But you have exactly the same difference between the groups, and you have a lot of variability. So you may be able to claim that this change is systematic. But then if a person comes with this abundance of the protein, you don't know if it's a healthy group or disease group. So this is not actionable. So this is an example where small p-value is not really predictive. Or here's another example, which is the opposite. So let's say we have two proteins, and now the blue dots are healthy subjects, and the red dots are the disease subjects. And we can see here that the proteins clearly change in abundance between conditions, and they're also predictive, because if you know what the protein is, you will know what the group membership is, right? But here is an example of another situation where the healthy subjects span the entire range of abundance of protein 1. But the disease subjects also span the entire range of abundance of protein 1. And so they will not be differentially abundant because on average their abundances do not change. The same thing for protein 2. But as a multivariate panel of two proteins, they're perfectly predictive, because if you know the combination of two proteins, you know exactly if it's a healthy or disease group. So this is an example where it's predictive but not regulated. And so my point is that different scientific objectives imply different types of statistical objectives and different types of statistical methods. And a lot of problems what I see, even at the design stage, even before you start the experiment, where people don't really match their scientific question to the statistical question. And this leads to kind of all the incorrect decisions later on. So the very first step we need to do before we start talking even about the experimental design is to agree what is the scientific goal and what is the corresponding statistical goal. And so just to finish, the third statistical goal is class prediction. So this is what would be necessary for biomarker discovery. And so the methods you would use here would be logistic regression, random forest, support vector machine, deep learning, whatever you would like to do, neural networks. So, there's a lot to choose from. And the readouts would be sensitivity, specificity, so the error rates of prediction, right, which t-test or ANOVA will not give to you. And the methods would go about like this. So let's say you have two proteins. The blue are the healthy subjects, red are the disease. So this is a decision rule, which essentially ignores protein 1. And it says large values of protein 2 means healthy, and low values means disease. And you get some misclassification here, but overall it's not too bad, right? Or we can have a more complicated rule, which involves both proteins. And now it's nonlinear, and we have fewer misclassifications. Or we can have something even more complicated, which totally accommodates every data point that we have. But this rule is probably too flexible, right? And it probably overfits our data. And maybe next time we have other subjects, maybe this rule will not be quite right for them. And so the goal of these methods really is to find this middle ground where your decision rule is complex, but not too complex, right? So that it does not overfit the samples. And so that's what these methods would do. So this is important to know. So what I will do for the rest actually of today, I will focus on this middle situation of the goal number 2, where we'll look for differences in abundance on average. And I will do this primarily because this is one of the most frequently asked questions in proteomic experiments. And this is also used even in clinical experiments as an initial stage of screening. For example, if you want to narrow down your targets to a smaller number, which can later be kind of analyzed like that to discover biomarkers. So we will focus on this part. Does it make sense? Okay, so now experimental design. So let's say we establish that we want to work on group comparisons, right? So we want to compare multiple groups, so the average protein abundance between conditions. So what are the important things to consider? So this is what happens in my mind when someone comes to us with this type of projects. Statisticians think a lot in terms of abstractions, right? And so we try to understand the underlying concepts, right? And then see how the data reflect these underlying concepts. So this is the abstraction which happens in my mind. So let's say we have two groups, healthy and disease, just again to keep it simple, but can be any other knockout and wild type, you can translate this to your own context. So let's say we have two populations which are comparable in respect to everything which can affect protein abundance, such as age, gender, lifestyle, other diseases, whatever else, right? So they're comparable in all the things which can affect protein abundance, except for the fact that these subjects are healthy and these subjects have the disease. So the only difference between the two groups is the status of the disease. And the abstraction we use is that for all practical purposes, these populations are infinitely large, or at least large enough that we cannot really interrogate every single subject in this population and say, okay, what's your protein abundance, right? So we have really large populations. So therefore, we need to take some subjects from this population and include them in the experiment. And we just said that there's a natural variation of protein abundance from one person to another. So if we are to repeat our experiment again, we will have some other subjects. They will have other protein abundance, right? Not exactly identical to our study. And so this is the first layer of variation, in that if we have to reproduce the experiment, we'll have some other protein abundances in our studies. But this is not the whole story, because we still don't know what the protein abundances are. All we know is that we included subjects in our experiment. What we do next, we actually quantify protein abundance in the subjects using the technology which for the moment still has some noise and some variation and some uncertainty. And so this is the second layer of variation, which is the technical or technological variation. We have two layers of variation, the biological variation and the technical variation. And our goal is now to see what we observe, because this is the only thing we observe, right? So we do not observe the population as an abstraction. We don't even observe the abundance of the protein in the subjects. What we observe is the data acquired in mass spectrometry. And the process of statistical inference is to take what we see and make conclusions about what we don't see, but what we're interested in. So taking the data and making conclusions about the unknowns. And this is really our goal. So when we do things such as hypothesis testing and referring and producing p-values and things like that, so what we are really talking about is what can we say about the underlying populations given what we actually see in our data. And so this type of things can have at least two problems. One is the bias and the other is inefficiency. Bias means that we are systematically wrong. So what our conclusions from what we see are systematically different from what we want to know. And bias is the worst possible situation because bias is reproducible. If you do something wrong, you will repeat it again, you will get the same result, and you will say, hey, I reproduced my, as Andy was just saying earlier, right? So you are precisely wrong. So we have to make sure that we avoid that. And the second problem is inefficiency, because our experiment can have too much variation and uncertainty that what we end up with in the end is really not informative enough about what we want to know. And so this is just an inefficient, too much noise, not too much variation, so it's not efficient. And the goal of statistical experimental design is to avoid bias and inefficiency, and this is what we do. I won't do much math, but I just want to introduce you the notation because it may help you read papers along the same line in the literature. The convention in statistics is that we use Greek letters for things that we do not observe. So here we assume that there is a mean protein abundance in a healthy population and a mean protein abundance in a diseased population. And so these are Greek letters. And then we select some subjects from the population, we include them in the study, and we use again these Greek letters, but now each individual person has their own protein abundance. But again, it's unknown, so it's again Greek letters. What we actually do see is the peak intensities, and now we may have multiple peptides of the protein, multiple transitions of the peptide, so we get multiple technical replicates, right? And so our goal would be to take these Latin letters, everything that we observe, summarize this, understand their statistical properties, and make conclusions about the differences between the constants in the population. And the bias means that what we see is systematically different from what we want to know. And inefficiency means that the variance associated with our readout is too large to be useful. And we can prevent these two problems by three fundamental principles of experimental design. And these principles exist for a very long time. I did not invent them. And they are well known in many areas of, any area which conducts experiments. But again, somehow every time there's a new technology which does things larger and better, we, for some reason, tend to forget about them. And so I would like to make sure that we review that. Sounds good? Okay, please stop me because I can talk. So the first fundamental principle of experimental design is replication. I hope I do not need to convince you that we need to have replicates, but let's just talk about this for a minute, right? So let's say we have two groups, healthy and disease, and we have one value of protein abundance for healthy and one subject for disease. And so we have this difference. So again, remember on the log scale, the difference is the ratio. So let's say the ratio on the original scale between these two values is 2.5. Now, is it systematic or is it an artifact of random variation? What do you think? Well, exactly. Who knows, right? You cannot say much because you don't know how much random variation is out there. And here's a situation where you have very little random variation in each group, so this is likely to be a systematic change, but this is exactly the same change between the groups, but the underlying variation is much larger, and this is more likely to be an artifact. So 2 points. One is that full change cutoffs make no sense whatsoever because they do not account for variability underlying these changes. And the second is we use replicates to understand how much variation is in the system. And if we work with multiple peptides or multiple proteins, some peptides can have more variation than others for biological or for technological reasons. And this is really important to take into account. So the same cutoff will not be meaningful for different peptides. And the other reason for using replicates is that, well, even if we're in this situation, if we can collect more replicates, we can be really, really sure in this mean. And if we collect more replicates here, we can be really, really sure in this mean. So we may be able to say that this is a systematic change, even if we have a lot of variability. So more replicates will allow us to gain confidence in the change, even though there's quite a bit of uncertainty in the data. And because we have two layers of variation, biological replicates and biological variation and technical variation, we have two types of replicates. We have biological replicates and then we have technical replicates. And I will probably not have much time to go into that, but let me give you the upshot. Biological replicates are the most important. Because let's say you have one person, you have many technical replicates from this person. So now you quantify this person really, really, really well. You did many, many replicates, but it's still one person. You still don't know how much person-to-person variation is in the population. So if you have a fixed number of runs, you'll be much better off to allocate each run to a different biological replicate than to spend runs on quantifying the same person very well. Well, that's the first thing. Okay? Now, the second fundamental principle is randomization. And that, I hope you know, but I'm still surprised how often I see that this is not done in practice. We need to make sure that we randomize all the steps of data acquisition and data processing, the sample acquisition and data processing. So let me just give you an example. So let's say we have, again, a simple situation. We have one feature per subject in the healthy group, one feature per subject in the disease group. And now it turns out that we cannot conduct the full experiment in one day, so we need to spread it over multiple days. And let's say that we have healthy subjects acquired on day 1 and day 2, and then disease subjects acquired on day 3 and day 4. And now we do have the difference, but this difference is due to at least two things. It can be due to the disease, but it can also be due to day of data acquisition. And let me tell you, day of data acquisition is much more likely. This is an example where an experiment is essentially wasted, because no amount of statistical, fanciness can really completely deconvolute the contribution of these two causes. On the other hand, what we can do, we can randomly assign each subject to a different day of data acquisition. And so what happens now is that each group has randomly different days. And especially if the sample size is relatively large, all the days will be kind of averaged out across both groups. So what happens then is that if you do have day-to-day variation, it will inflate the overall variation in each group, because now you have not only the biological variation, but also day-to-day variation in your data. But at least you have no bias, right? Because this is bias due to the confounding of the day and disease. Now here you have inefficiency, but at least you have no bias. And so potentially if you have a large number of replicates, you can still claim that there is a difference. Randomization is especially useful because it protects us against biases from sources we're not aware of. If we don't even know what else can potentially affect our results, by randomizing, you essentially average over anything if you know it or you don't know it. And this is really important. And again, because we have two layers of variation, biological variation and technical variation, we have randomization of these two layers. And sometimes people only remember one layer. Maybe they randomize how the samples were, allocated to mass spectrometry measurements, but they do not randomize upstream steps, or they do not randomize other things. And this is also very important. Some examples of that. So this is a manuscript which I appreciate very much. It's old, but I think it still makes really useful points. So these are statisticians from MD Anderson Cancer Center, and they do a lot of service work where they help cancer researchers design and analyze their experiments. And this was also, it was still working with CELDI workflows. But what they did, they published a manuscript where they showed examples how things can go wrong. in their practical experience. So this was an example where they had 5 types of cancer, so five groups. They had the data, was sold it off, they did all the right things, normalized, picked, and so on. The data arrived to them blinded, so they didn't know which sample comes from which disease group. And of course, if you don't know any class membership, what you do? Class discovery, right? So they did this type of clustering of samples, and they were excited to see that they found 1, 2, 3, 4, 5, 6 disease groups instead of 5. So they said, yay, maybe we found a cancer subtype from the cell experiments. But then the next thing they did was they had exactly the same plot where the x-axis is the order of the samples in this dendrogram, but the y-axis is the date of sample acquisition. And so you see that those clusters match perfectly the date of data collection. And later it turned out that some of the groups were acquired earlier than other groups, so this is an example of an experiment which was a waste of money and time. But it's good to know that they really try to emphasize the importance of this, and they published this type of examples. So this is one. Well, okay, fine, right? Didn't go very far, except yes.
Okay, so you should randomize samples while sample preparation and analysis. But should you do it multiple times throughout the process as well? I mean, you prepare samples in a plate. You randomize the samples in the beginning, you put them on a plate separately.
It's often a good idea.
And then you follow with the same layout throughout the sample prep.
It's often a good idea if it's practical. Yeah. So at least once for sure. For example, if you have a streamlined, like robots and so on, it may be difficult to rearrange that. But generally, it's a really good idea, especially because there is some time going by between sample preparation and data acquisition. And during this time, just imagine, right, if you have, depending like multiple plates, right, and how they're sitting, somewhere, right, artifacts can be added after sample processing, right? And so you want to randomize them as well. So if it's very streamlined in one step, you probably don't need to do this. But if time happens between different steps, then it's really a good idea to randomize in between, just to prevent whatever. So I'm not quite sure about kind of the details, but you can imagine that, for example, storage can affect, right, things which are sitting kind of outside of the storage place versus inside, right, can affect things. So secondary randomization will protect against that. I don't know if you know what I mean. Okay. So to summarize, if it's one step very streamlined, once it's enough. If time occurs between multiple steps, it's a really good idea to randomize. Okay, but this is another example which is much worse in that it actually made it to a publication. So this was a Nature Genetics publication in genomics, which I took from the blog of a statistician colleague from Johns Hopkins. These researchers studied gene expression between Asians and Europeans. And they published a manuscript where they found that 78% of genes are differentially expressed between Europeans and Asians. Let me repeat, 78% of genes are differentially expressed between Europeans and Asians.
It's like calling your species.
Exactly, right? And they were stunned just as you are. And they decided to look into this more and reproduce this experiment. The good thing is that the data were actually available in associated with the study, and they looked at the data analysis, they looked at all the procedures, they didn't find anything wrong with that. They felt like it was a reasonable way to analyze the data, until the time when they decided to look at the very raw data, and this was the time where they used Affymetrix chips. And in Affymetrix chips in the corner, there's a timestamp. The time when the samples were hybridized. And what they found was that there was a 1-year difference between the time when they collected data on gene expression from Asians and from Europeans. So no wonder 78% of things changed, because it was a lot of time in between. And the good thing is that you could find this in the data, right? The bad thing is that the researchers didn't even think about the problems associated with that. I don't know what happened with the paper, but it was last time when they were looking at that, it was still around. You really don't want that. Okay. And this is just one of the kind of very frequently used quotes. To call in the statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment died of. So this is exactly the case, because all you can explain now why it's wrong, but there is no way you can rescue this experiment.
There's an interesting story here. It's that it was two faculty members in our department here who actually wrote the technical correspondence about this paper.
Oh, really?
So it was John Story and Josh Akey that the reason why they got kind of frustrated with it is because they did an almost identical experiment but between Caucasians and Africans, and they saw almost very little difference. And they said, how can there be this enormous discrepancy between other ethnic groups? And that's why they caused to dig in the data pretty carefully.
Yeah. But it took, so there is a term now, they have forensic statisticians, right, where you have to go and dig into, let's not get our experiments to that point. Okay, so this is what is happening with randomization. And this is really important, but as we mentioned earlier, if we do randomize, it does inflate the variability in our groups, because now we add on top of the biological variation, the variation between different instances of data collection and sample processing. The third principle of experimental design is blocking, and I would say that this is the one which requires most statistical expertise. So in terms of acquiring replicates and randomizing, I think you can do this. So you can do, for example, a random sequence in Excel or in R. By the way, don't do it by hand. Humans are really terrible at randomization. So I read a story somewhere where they were assigning some treatments to mice, and so they would just essentially grab mice from the cage and assign them a treatment, and then the remaining were like controls or something like that. Turns out the mice they couldn't grab were slower. And this impacted their conclusions in some ways. I don't remember the details, but humans are really terrible at this type of thing, so you really want to do it at least in Excel or in R and in some other tools, and make sure that you allocate these labels. But in addition to saving the labels, you need to save the protocol, like how you randomized. And this is important because if you tell me, okay, I do 1, 15, 3, 7, is it okay? Like, I don't know. It depends how these numbers were obtained in the first place. So it's also important to write down exactly what was done to arrive to these numbers. All right, but now blocking is something which really requires us to understand the structure of variation, the structure of experiment a lot more. And it helps us reduce both bias and inefficiency. It is useful when there is a source of variation which impacts the abundance of the proteins and we know about it. So if we don't know, there's not much we can do, we just have to randomize. But if we know, for example, we have a day-to-day variation, that we cannot help it, but we know it exists, then blocking can help us improve the quality of our conclusions. And so this was just continue on this example where we have multiple days, right? And so if we compare the mean of healthy to the mean of the disease, we may have too much variation to be able to make meaningful conclusions. But what we can do instead, we can impose a restriction on randomization. So a technical definition of blocking is restriction on randomization. You don't randomize things completely, but you impose restrictions. In this case, we impose a restriction that on every day, we want to have one healthy and one disease. So not just randomly taking samples from the two groups, but taking one healthy randomly and then one disease randomly. Day 2, we take again one healthy, one disease. Day 3, one healthy and one disease. And so now if you look at this, so these are pairs which are acquired in each day, and you can see that clearly there's a day-to-day variation. But then if you compare within the same day, you would realize that the differences all systematically point in the same direction. And this means that this is evidence that in disease group, the protein is upregulated as compared to the healthy group. Now, I do one healthy and one disease on each day just because I can make the picture here. But there is no restriction to have only one, you can have a lot more. And there are statistical methods which allow you to both allocate samples to these blocks and also to analyze the samples. It will be not just the difference, but it will be more generalized approach, but they're very standard statistical methods based on analysis of variance and linear models which can analyze blocks of a larger size. We look here at the differences between groups. Now, we had this conversation about internal standards this morning. So imagine now that this group is actually not healthy, but they are internal standards. This is exactly what we are doing. We are looking at the deviations on the log scale from the internal standards. So ratios on the original scale. That's exactly, so using the internal standards in the same run with endogenous peptide is one instance of blocking in statistical language. And we can use this group as internal standards, but we can also use this as a different disease group. And we can still look at the averages within each disease group. And this is the case where we can accomplish two things. One is that if we have very small sample size, if we completely randomize, we can still just by random chance end up with more healthy on day 1 and more disease on day 2 or something like that. This would systematically make sure that every group is exactly in the same day. And then if we analyze this accordingly, we also gain efficiency because we essentially eliminate the day-to-day variation analytically. Another example of this block will be SILAC, for example, if you have two groups within the same sample. So that's now within the run, it will be this type of blocking. But let me just for the moment focus on the day here, right? So so it requires two things. One is understanding that you have a source of variation that exists, you cannot control. You need to allocate your resources accordingly. And then second, we need to analyze the data differently to account for that. For example, here, if I decided to compare the mean of this group to the mean of this group, I could still do this and I would not have the bias because I have the same number of subjects in each day, but I would miss the opportunity to reduce the variation because I still have to work with the day-to-day variation. But if we now look at differences between the groups, I could eliminate the day-to-day variation. This is a simple example. When things become more complicated, this is where you really would benefit from statistical expertise. What this means is that in many experiments, when it's not entirely obvious how to conduct them, your best way to go is to talk to a statistician before collecting the data. And if nothing else makes sense today, this is the message I really wanted to remember. Talk to a statistician before collecting the data. Everything else will be fine. And because we have two layers of variation, remember biological and technical variation, we have two types of blocking, biological blocking on biological factors and blocking on technological factors. And we need to work with that. And just give you a little bit of examples. So this was one example from the same paper where it was another experiment, also cell off. They also did the right things and did the clustering, had two groups. They were happy because they had two types of cancer. When they blinded that, they realized that the two clusters were driven by the protocol change in the middle of the experiment. Well, no wonder, right? It's probably obvious. Now, if experiments are very long running, it may be necessary to change the protocol just because of the constraint. Maybe change the batch of the material, right? Or maybe you have a service appointment scheduled in the middle, or who knows, right? If these things are known, you can plan with that. So what would be a good solution here is that you would have the same number of healthy and disease before the protocol change and the same number of healthy and disease after the protocol change. And you would essentially look at each batch and then you would integrate the differences. And it is possible to work with that, right? If they didn't do this, if these were all healthy and these are all disease, again, this is completely wasted. Just to emphasize a little bit more on the design, so as I just said, we have two types of variation, so two types of blocking. The blocking with respect to biological factors is referred as matching. And this is something which is very much used in clinical studies especially. I think it's probably less of an issue in work with model organisms, but it's definitely important with human subjects. And the idea is that, remember we said the populations are very large and they have different things which can affect protein abundance, and age and gender always affect everything, so at the very least, right? And if your number of subjects in the study is fairly small, you may still, even if you randomly select subjects and do everything correctly, you may still end up with, let's say, healthy male and old, and then disease female and young, or something like that. You want to prevent that because then you have differences between the groups others than the disease. And what you can do, you can impose again restriction randomization and say, okay, I want one younger female from each group. Okay, randomly selected. Now I want one older female from each group, randomly selected. Now I want one younger male from each group, and so on. Now you may have, here for example, we have more kind of older female than younger female. It's okay as long as we have the same numbers in each of the disease groups. So the important thing is that the rows match exactly. And then now, if we look at the differences between red and blue, they have exactly the same confounding factors. And now the difference that we find will be associated with the disease more likely than with other confounding factors. And just to give you an example how we did one of the studies like that a while ago, this was a collaboration with Munich Heart Center, where researchers collected plasma samples from everybody who would come to their clinic for treatment, and they essentially stored them. So they had a repository of these plasma samples, and then they wanted to select a sample, just a smaller subset, just for an initial kind of screen and a pilot study. And the clinicians said that they wanted to stratify it by age and gender. That was the cutoff that they told us would make sense. And these were the number of samples they had in the freezer. And then the idea would be, if you really want to match, so block by the clinical factors, what you want to do is to have the same number of replicates in each row. So if you take older female, it will be the same number across all the disease groups. And then you have older male, again, the same number of replicates across the disease group. And so that's the type of thing that you can do. It doesn't matter if some rows have more than others, as long as every condition in a row has the same number of replicates. So that's the idea. Unfortunately, I should have included the specific slide on the internal standards, but I hope you see by now as well, right, that if we look at the differences between the endogenous and internal standards on the log scale, it allows us to reduce the variation, just like Andy was telling us this morning. Mm-hmm.
Since we are dealing with the average and the sample deviation for each row, why we should have the same number of replications?
Well, so imagine now that you have, like here, so you have group 1 and they're all male and older, right? And here they are female and younger. What you will be doing, you will take protein abundance among these subjects and compare it to protein abundance among these subjects.
I'm asking about the numbers between different types of, for example, the control and different types of the
Here?
disease.
Mm-hmm.
And these suggest us to have the same numbers
Correct.
for all of these groups.
Correct.
What is the reason for that? Because statistic, we are dealing with average.
No, nothing to do with how we analyze the data. It's really the origin of the protein abundances that you see. It's the property of your data. So imagine, well, same thing, right? So what you will be doing, you will be comparing protein abundances among these subjects to protein abundance among these subjects. If in this group you have all older female and in this group you have all younger male, the difference can be due to the disease, but also to the age and gender. And you really want to be able to claim that the difference is due to the disease and not to other sources. And by doing this systematically, you now make sure that the subjects have the same property, so the same distributions of risk factors or age or anything else which can affect protein abundance in addition to the status of the disease. Because your point is to study the disease, right, and not study the age. If you wanted to study age, then you would be doing something else. Does it make sense? You really want to be able to say, I'm comparing healthy and disease, and the difference in protein abundance is associated with the disease, and not because these are young and these are old. These are biological things which affect protein abundance just as much as technological things can affect protein abundance. And from what I'm hearing from my clinical collaborators, age and gender affect everything. So you can pretty much expect upfront that people of different age groups and different gender, there will be some effects which will confound what you really want to study as disease versus healthy.
I think sometimes, most of the time for clinical sample, we are limited to the number of clinical samples for example, specific cancer. The sample is really precious and we have just limited number of the cancer samples, but we can actually find lots of controls.
Correct.
We can find
So what you can do then, well, you pretty much build the same table here, right, from what you have. You say, here's my control group. Let me account for things which can affect protein abundance besides the disease. And maybe, I don't know, maybe some other diseases, maybe treatments, right? Maybe, I don't know, lifestyle, like smoking, I don't know, whatever, right? And then you say, in each group, how many disease samples I have. Now, how many controls I have. You can actually double what I didn't show here, but you can have 30, 30, 20, and 20. So the total number of subjects in each group doesn't have to be identical. But the distribution of risk factors in each group has to be the same. Yeah, so you could totally have 3, 3, 2, 2, and then 30, 30, 20, 20. Yeah.
If you were to analyze the entire sample,
This?
top one,
Mm-hmm.
how would you divide up the groups? Let's say you have to do it in 10 different batches. So what design would you use for that?
If you wanted to analyze this, well, first of all, you see that the distributions differ quite a bit, right? It does look like this group has more than others, right? So what I would probably do is to make sure I would minimize the differences between the groups, right? So it's always the same thing. First select the subjects and then allocate them to data collection steps, right? So first you would say, okay, what are the subjects which represent what I want to study in the best possible way? And then I would say, okay, I have that many groups, I have that many subjects per group. Now I need to allocate the subjects to the data sample processing and spectral acquisition. And depending on what is going on, you would probably want to have some block randomization. And for example, let's say a day of data collection is a proxy for many things, right? So let's say we would want to block on the day, right? So within one day, you would make sure you have the same number of every disease group. If one disease group is much larger than another, so you keep kind of the proportions of that, right? Now the next day, again, either exactly the same number of healthy disease or the same proportions in this day. So now, for example, if one day doesn't work out for some reason, you lose exactly the same proportion of everything, right? You do not lose disproportionately healthy or disproportionately disease. So this is one way to do it. Another way, if you have like a continuous process, which is like automated, you don't like have these day blocks. What you can do also is what is called the minimal replicate set. In this case, minimal replicate set will have one subject from each group. Or if you have one group much larger than the other, you will have more in one group, but you have the same representatives from each group. So what I would do is I would take representatives from each group, shuffle. This is my one replicate set. Then again, I will take the same number of representatives from each group, shuffle, right? And so on. So now I don't have like really blocks kind of clearly defined, but I'm just dividing my time in a way that things I want to compare are the closest in time. And then if something happens in the middle of the project or at the end of the project, in the beginning, you still have all the comparisons you want to use. So this is the best way to do it. But this is simple. But now imagine you have additional complication in sample processing, right, and other protocols. So again, getting statistical expertise would be quite useful.
So what about doing multivariate business analysis afterwards and finding the co-factors or co-variables which are affecting like feeding together weights afterwards instead of doing these kind of situations before the process? How effective is that?
It is not. Especially in the type of experiments where we talk about, because we have a very large number of proteins. Even if we do just SRM experiments, right, we still have several hundred proteins, right? If the number of replicates is relatively small compared to the number of proteins, if you try to find things which affect age or gender affecting different proteins, you will always find something just by random chance, because something which would appear associated. You know, if you look hard enough, you will find something, right? So here looking hard enough means more and more proteins, right? So you always find one protein, oh, look, for this protein, age seems to matter, right? And then for next protein, oh, gender seems to matter, right? You are much better off understanding this in advance, especially in targeted workflows where you're already doing kind of, you're not doing discovery, you're already trying to kind of go more deeper in the science and account for this as part of your protocol. If you think that age and gender are appropriate, you can definitely adjust for them. But you will adjust for them best, again, if they're equally distributed between the groups. Because at that point, age and gender become your blocks, right? And you can also, so like in my picture here, what you could do, you could look for differences in protein abundance in this group, differences in this group, right? And then combine them. And then you would eliminate the age and gender variation, which you're not interested in. But this would mean, again, multiple things. First, understanding that these affect enough proteins, right? It would require collecting the data like that, and it would require to adjust your sample size so that you have enough data to actually add for this. So all of these adjustments typically require larger sample size. And they're a good thing to do, but it is extremely important that you plan ahead for that. And the discovery and associations using multivariate analysis kind of post hoc, as I say, in this type of experiments, you are very likely to get some associations which just come out because you have many proteins to look at. And if you start working with DIA experiments, well then you're guaranteed to find all kinds of associations there, right? And it's fine, you can still do this, right? But then it cannot be part of your confirmatory step. It has to be part of your exploratory step, which you would have to validate later. But for experiments which already go kind of trying to understand this is the association between protein abundance and the disease. These are the proteins which are regulated in the disease. You can definitely account for age and gender, but you need to plan in advance as opposed to post fact. Okay. So this is really for the design. So just to kind of summarize here, randomization, replication randomization, this is something which is easy to do even if you are kind of on your own. But these advanced methods which increase the effectiveness of the experiment, minimize the bias, it is best to talk to a statistician and design this together, because you need to design the experiment and then analyze this accordingly to the design. And you best design the experiment when you know how it will be analyzed later.
Do you have any limitation for blocking? For example, if I know I have 4 different factors affecting my results, I can have 4 different plots, 5 plots. Is there any limitation for this number?
Just the sample size. So the more adjustments you want to do, the more replicates you would need to make sure that you can still estimate. So, for example, if this was my example with blocking, right? You start with 8 data points, but your 8 data points become 4 pairs. So now suddenly you have 4 pairs as opposed to 8 data points. Now let's say some of this you want to block on something else. So instead of 4 pairs, maybe you will end up with 2 pairs of pairs. And the more you want to adjust for, the more sample size you need to make sure that you can still estimate the remaining variation.
In terms of the statistical analysis, we don't have any limitation.
Well, sample size, right? Because if you block enough, then you just have one unique pair. So, for example, if you want to have, compare healthy and diseased younger female acquired on Monday, well, you only have one pair acquired on Monday, right? And so you don't know how much those vary, right? So to make sure that you can do this, you need to have multiple pairs acquired on Monday, meaning larger sample size, right? Mm-hmm.
How does the experimental design changes between the targeted dynamics and some classical DBA like transmission dynamics? I mean, do you follow the same procedures to design?
Generally, yes. The difference would be in the details, such as sample size calculation, because the variability associated with DDA versus SRM, and also the number of proteins that you quantify, have implications on the sample size. But the concepts we're talking about are the same concepts. So you still need to randomize, you still need to, replicate, and you still need to block. Usually, if you have, and actually, I'm just looking at the time, I feel like, I hope I can talk to you about that. But usually, if you have less technological variation, you would need smaller sample size. And if you have fewer proteins, you will need a relatively small sample size. So in particular for DIA experiments, you would need more replicates. That's my experience. Yeah. Let's see if we can get to that. I really want you to hear about this at least a little bit. Okay, let me... Right, so if I don't talk about it now, I'll talk about this after lunch. So I'll make sure I'll get back to this point. But let me now tell you about the basic data analysis, because we will be using it later in the afternoon. So I want to make sure we're on the same page as far as like things such as, t-test, hypothesis testing, t-test, and p-values. Okay, so basic data analysis. Remember, just to refresh your memory, right, we have these different concepts, and what we observe is actually peak intensities, and we summarize them and use them to make conclusions about the unknowns. And our goal is to kind of minimize the bias and inefficiency, but to do this, we also need to understand what are the statistical properties of the things which we actually observe. And the statistical properties are characterized by a statistical model. And conclusions regarding the unknowns, they're called model-based inference, because we specify some abstractions, make some assumptions which allow us to make conclusions about what we don't see. Okay, very simple case. Let's say we have two groups and just one feature per protein per sample, label-free, right? Just to keep it very simple. And we want to compare the replicates in the healthy group to the disease group, so on average. This is just a slide to summarize what happens when we work on the log scale as opposed to on the original scale. Fold change. We refer to this to say, well, there's some typical value, some kind of consensus value in group 1 divided by some typical consensus value in group 2. Now, if we take the log of the ratio is the difference of logs, right? So that's what we have. But now, this is, so how does that relate to replicates? Because these are just typical or consensus values. This is just to kind of show this to you in more detail. What we want to do, right? We look at the differences between the summaries of two groups. So this is our estimates of the log fold change. Now I can just open it up. I promise not much more math than that. So this is just the arithmetic mean of the log intensities, right? Minus the arithmetic mean of the log intensities. Now, I just put it explicitly that this is the log intensity, right? So this is now the log of the original values. But now log has this property that sum of the log is the log of a product. And so we can put it back like this, and we can put it back like this. So essentially what it means is that if we summarize our data across replicas by averages on the log scale, our typical values in each group, they are geometric means on the original scale. Just so you know. So we're working on the log scale, and it's always easier for us because from the statistical perspective, it's always easier to work with sums and differences as opposed to ratios and products. But when we do this and we work on the log scale, it means that on the original scale, we're actually talking about geometric means in each group. This is just to kind of, so you know. Okay, now we want to know if there is a difference between the two groups on average over replicates. And again, remember our quantity of interest is actually the means in the population. What we observe is our data. We want to know if in the population the means change between the groups. And because we never observe them, we will never, ever know what they are, right? So we will never know what the difference between the means and the population is. But we can make guesses. And the way hypothesis testing, frequentist hypothesis testing works in statistics is that we will make a guess about the difference between the two groups, and we will say, let's say it's 0. So this is the case we're not interested in. It's a status quo, kind of, something we're not interested in. So let's say it is 0. And now the alternative would be that it's something else other than 0. And what we'll now need to do is to construct this quantity, which is essentially a signal-to-noise ratio. The signal is the difference between the groups. So again, our consensus kind of summaries between the two groups. And the noise is the estimate of variation associated with the signal. And the estimate of variation has this particular form. Now, I will spend a few minutes on that. the S squared is just sample variance in this group, S1, right? And S2 squared is sample variance in this group. This is easy. But what we also do, we divide them by the sample size. And this quantity sample variance divided by the sample size, if we take the square root of this, it is called standard error of the mean. And this is very important. I really want to emphasize the difference between standard deviation and standard error. So the square root of this is standard deviation. The square root of this quantity divided by n is the standard error. And they reflect fairly different things. So standard deviation tells us how much uncertainty we have in the population. Standard error tells us how much uncertainty we have in the mean of our data. And you see that the sample size here is in denominator. It means that the larger the sample size, the less uncertainty we have in the mean. But we can still have uncertainty in the population. Just to give you an illustration of that, I have several figures from these vignettes called points of significance in nature methods. They're very short vignettes, like 2 pages, focused on some specific topics in statistics. And so I usually take figures from that because there's also text that goes with them. You can go to these vignettes and read more about that. They illustrate this concept of standard error. Let's say we have our population which has roughly normal distribution of protein abundances between the groups. Now what we do here, let's say we have 3 subjects from this population. Calculate the mean. Now go back, take 3 other subjects from this population. Calculate the mean. Then go back, take 3 more subjects. Calculate the mean. So here is the histogram of those means of 3 subjects. And you see already that this histogram is narrower than the original histogram, right? Because the means, they're much more precise, right? They're more oriented towards the center. Now we can do the same thing, but instead of 3 subjects, take 5 subjects, or 10 or 20. And you can see that the more subjects we take, our histograms become narrower and narrower. So there's less and less variability in the mean from one instance of collecting the data to another instance of collecting the data. So the width of this histogram is the standard error of the mean. The width of this histogram is the standard deviation of the population. Right? So the standard deviation describes the population. It will never get smaller because it's what we're studying. Standard error will get smaller and smaller if we have more and more data. And that's what I meant when I said we're really, really confident in the mean. If we have more data, well, our mean will be more and more reproducible. And this would allow us to claim systematic changes, right, if we have more replicates. Now, the interesting thing is that it works the same way also, even if our data are not normally distributed. So we can start from all kinds of weird distributions, but if we repeat the same process, the histograms of the means, they're also more and more bell-shaped distributed, and they're also becoming narrow and narrow. They're not narrow as here if we start from some nice case, but eventually they will also become much smaller and more bell-shaped. This is called the central limit theorem, that our means are more and more narrow and more bell-shaped distributed. And this is important for us because this would allow us to essentially make statements about the populations if we use this type of framework. Okay, so now going back, what happens, right? So here we have our signal, difference between the 2 groups. So this is essentially the variance of the mean in the first group. This is the variance of the mean in the second group. We combine them because we want to look at the 2 groups. We take square root to put them on the same scale as the original data. So this is our signal-to-noise ratio. The signal is the difference between the groups. The noise is how much variation we have associated with this difference. Okay, so that's what we need to do, right? So this is now our kind of summary of the data. Remember I was saying earlier that we take some summaries, right? And now we need to describe their properties to make conclusions about the unknowns. So that's what we are working towards. Now, the last thing that we need to ask, well, is how large has to be the signal to noise to claim that the signal is larger than noise, right? So essentially the change between the groups is larger than the underlying variation in the means. And this is the last piece. So if we assume that the difference between the means is 0, And if we assume that the data follow normal distributions, and if we construct the signal to noise as we just did, then we can analytically, without any data, just purely mathematically, say what would be the expected range of the signal to noise values. And by range, I can mean more probability distribution. So it looks, again, a normal distribution, very kind of bell-shaped. Alternatively, if our data are not normal, but the sample size is large, we can claim the central limit theorem, and we will say that this is another distribution called student distribution, which is also bell-shaped, which is very similar to normal. So in both cases, either if our data originally are normal or not normal, we can say mathematically what would be the probability distribution of the signal-to-noise ratio. So essentially, this is our expected range of the signal-to-noise ratios, provided that there is no difference between the two groups. This is part of the assumption. And also there's another assumption that data are independent. Now what we will do next, so this is something we determine mathematically, right? Now what we do next, we actually can calculate the signal-to-noise ratio because we have our data. And what we will do, we will overlay this on top of this distribution. Now, if we overlay this on top and it falls in this middle kind of range, we'll say, yeah, well, that's what we expected, right? Therefore, there is no evidence against our initial claim that there is no difference between the two groups. On the other hand, if we overlay the signal to noise on top and it's really in the extreme somewhere, either in this tail or in this tail, we say, wait a minute, it's very unusual. It's too large or too small compared to what we would expect. What's wrong? Well, what's wrong is that we assume that there's no difference between the two groups. So it's evidence against this and in favor of the alternative. And so the very last piece is that, you know, these probability distributions, they work in a way that the area under the curve is the probability of this particular range. So we can find this cutoff, such as the area to the left plus the area to the right is some probability that we want to have, let's say, 1% or 10%. So this is the false positive rate, and this determines essentially the decisions. And we say if our calculated signal to noise is in this tail, on this tail, outside of this range, then we reject the null hypothesis. Otherwise, there is no evidence. So we never accept null hypothesis, we just say there is no evidence against the null hypothesis. Because maybe if we had more data, maybe then our signal to noise will be larger. So this is how hypothesis testing works, right? So we make this assumption. We use this data representation to establish the expected range of the signal to noise ratios, and it allows us to see if our assumption has evidence against our assumption. And the last related kind of concept with this is the p-value. So we can actually overlay the signal to noise here, and the p-value will be the area under the curve to the left of the observed and to the right, because it's symmetric, so usually they look at the symmetric tails, right? And so, of course, if the p-value is less than our alpha, we reject the null hypothesis because it's the same thing, right? So if the area to the left plus the area to the right is smaller than the red area, it means that we are in the rejection region. So we reject the null hypothesis. It's just a equivalent way of looking at the same thing. So that's it. Does that make sense?
So, as you said, when you transform data, for example, to log, I mean, I think you usually assume that so, but does it make a difference if you perform just t-test with log data or?
So everything we do on the log scale, because on the log scale, the data are much closer to the normal distribution than on the original scale. On the original scale, it would be more skewed, right? And so on the original scale, usually things are multiplicative, right? So you want to work with ratios, and ratios are more difficult to work with in terms of establishing the properties of that. So sums and differences are much easier. That's why we work with them, because we can actually come up with these distributions for them. Now, you can also claim central limit theorem and say I'm starting with something skewed, and I would still be doing a t-test on that. But you can see here that it will take more replicates to really, for the central limit theorem to kick in. So if you do it on the original scale, it would probably require more replicates to be correct. In our practice, we always log transform, and we assume that things are additive so we can look at the differences on the log scale. But then at the very end, if fold changes are meaningful to you, we essentially use this to transform back on the fold chain scale.
So then your final conclusion will be based on, I mean, technically we'll be saying that there is no significant fold change, but perhaps it's not.
No, what you will see is that I reject the null hypothesis, and the estimated fold change is that. So your hypothesis testing occurs on the log scale, but you can still report all kinds of summaries for this protein, including fold change. But the fold change would be by looking at the geometric mean in group 1 versus geometric mean in group 2.
So you have to compare, you have to say it's fold change and not absolute value.
Oh, there's no absolute value, right? Because we don't do absolute quantification here. Everything is relative here. Yeah. Okay. So now I really want to talk a little bit about statistical power. So maybe this would be a little bit going kind of towards this question, SRM versus DDA versus DIA, how it affects the sample size calculation. So this is quite important. So okay, what did we establish? So we have some summary which I call signal to noise. In statistical language, it's called a test statistic, but doesn't matter. We just summary of the data, right? We can analytically establish the probability distribution that it would have if there was no difference between the groups. Now we will overlay what we actually observe and see if it's extreme or not, right? So that's what we would do. This is just another representation from this vignette, same thing, right? And so, by the way, sometimes people only concentrate on one tail when they know for a fact just from the biology that it can only be upregulated and not downregulated in one group. Then they put all the alpha in one tail. These are called one-sided tests. So very simple extension. So here, their example focuses on this one-sided test. They put all the alpha on one tail, so they do not consider possibility of downregulation. And this is my signal-to-noise ratio, right? So this is what would happen if the null hypothesis is true. So if there is no difference between the groups. And in reality, if the protein is really regulated, this signal-to-noise ratio, well, let's say group 1 has larger protein abundance than group 2. So this signal-to-noise will tend to have values which are larger than 0, right? It will fluctuate somewhere around some positive values. So in reality, this quantity will be shifted to the right, its distribution, right? And so if this is my decision, and I reject the null hypothesis based on this cutoff, in reality, this is the probability to reject the null hypothesis, right? Because this probability is calculated based on the assumption that there is no change. If in reality there is a change, everything shifts. And so in reality, your probability to reject the null hypothesis is much larger. It is this. And this is called the power of the hypothesis testing. And this power depends on how far to the right your signal to noise shifted compared to the null distribution. Okay, and so here, for example, it shifted a little bit, so now you have the area, the blue area here is 0.64, or it can shift even more, and now the area for the same decision, right, the area is 0.8, so here the power is 0.8, and here the power is 0.64. Okay? Now, what gives us statistical power? It's essentially anything which will take this quantity and will shift it to the right, make it larger. So what can this be? Well, if the difference between the groups is very large, well, this difference in observed values will also tend to be larger, right? So clearly, the more regulation there is, right, the more change occurs, the more this will shift to the right, and the more it's easy to reject the null hypothesis, right? So that's very clear. But there are other things too, right? So another way to shift this whole thing to the right is to reduce the denominator. Now, the denominator can be reduced in at least two ways. One is reduce the variance associated with each measurement group, right? So some of this is biological variance, but some of this is technological variance. So reducing the measurement error will give you more power to reject the null hypothesis, right? And the third thing is to increase the sample size, right? It's in a denominator of denominator. So larger sample size would mean larger overall quantity. And so increasing sample size will also move the whole thing to the right, and it will also give you more statistical power. And so these are the implications in terms of sample size, and you essentially work with that to do sample size calculations. So you specify, you fix alpha, you say how much power you want to have, and you work with that to establish what are the sample size that you will have. But for that, what you would need is to have some sense of how much variability you have in your experiment, so you need to have some pilot study which quantifies the variability in your biological system. Okay? And therefore, when people come to me and say, how many replicates do I need? Can I use 3 patients? I don't know if you have seen this video of experimentalists talking to a statistician. But my grant is due tomorrow. Can I use 3 patients? I don't know, because it really depends on how much variation we have and how much regulation we have. And the sample size will be determined by how much power you want to have, how much numerator you have, and how much denominator you have. So these calculations are very experiment specific and very specific to a biological system and also very specific to a technological measurement system. And of course, another way to increase the power is to give yourself more error, right? So this is what people sometimes do. Instead of using 5% false positive rate, they have more. Of course, you will increase the power, but this is not a good way to go about this because it's the same data, right? So you just have more false positives. So that's not a good idea. Okay? All right. So an alternative way to do, not alternative, but a complementary way of doing hypothesis testing is to also focus not only on p-values and on the conclusions, reject and not reject the null hypothesis, but also look at the confidence intervals. And this is because p-values, well, it's nice because it's very clear cutoff, right? 0.05 or 0.1, and you know if it's rejecting or not rejecting, but it doesn't really tell you the magnitude of change, it doesn't really tell you the directionality of change. It just tells you that it's significant. Confidence intervals can give you more information like that, and so it's a very nice complementary way of doing this. Now, confidence intervals, they're not actually intervals, they're procedures. So a confidence interval is actually a procedure which has some properties. So, for example, if we want to have a 95% confidence interval for a mean, it's a procedure which gives you the left and right bound of the interval, such that if you repeat this procedure over and over, you keep collecting the data, repeating this procedure, 95 intervals out of 100 will contain the true value. And 5 out of 100 intervals will actually miss the true value. So once again, right, it's a procedure which determines lower and upper bound. If we collect 100 data sets and apply the same procedure, 95 data sets will contain the true unknown value, but 5 data sets will miss the true unknown value. And how do we know what are the right formulas essentially for the lower and upper confidence interval? Well, it's still based on the same signal to noise that we just talked about and the fact that we can analytically derive their properties. So you see here we have the bounds of confidence interval, they're exactly our signals. Here we have exactly our variability, and those multipliers, they're actually critical values which give us the decision rules for the hypothesis testing. So they're exactly the same value. So if you have a confidence interval for the difference which includes 0, it's equivalent to not rejecting the null hypothesis. So it's the same procedure, just represents the results of this procedure in a different way. And so now what it means is that we have at least 3 ways of representing our results, actually 4 ways representing our results. So one way is hypothesis testing, right? Pass or fail, reject null hypothesis or not. But the other 3 ways is to plot our estimates of means in one group versus in another group with error bars. And we now have 3 types of error bars. We have standard deviations, we have standard errors for the mean, and we have confidence intervals. And they're very different. And so what I see a lot in papers is people say, here is my whatever difference between the groups and the error bar. Like, well, what is this error bar? Depending on what this error bar is, it may have very different conclusions from this and very different interpretations. And these vignettes highlight that. So for example, here, for a single group in black, you have 95% confidence interval. In gray, you have a standard error of the mean. And the y-axis is the sample size. Well, clearly, the sample size is larger means narrow and narrow intervals, but you also see the standard errors for the mean. They're much smaller than the intervals, the confidence intervals. And standard errors for the mean, they don't have any probabilistic interpretation. You don't know how often they will be right. but the confidence intervals have probabilistic interpretation. You say 95 times out of 100, it will contain the true value. But the appearance is very different, and the conclusions will be very different. And of course, you can see here the sample size is in the denominator. This is why they become narrower and narrower as sample size increases. And this is just an illustration also from these vignettes. Here, they take 10 replicates, and they adjust the variability in two groups. So one group has mean 0, the other group has mean 1, just arbitrary. And they adjust variability such that 3 types of error bars look identical. And you see that the conclusions for hypothesis testing are very different. The p-values are very different from these 3 identical looking error bars, depending on which error bar it is. And here is the other way around. They fix the p-values, and they show how different looking error bars will be, depending on which error bar it is. So again, it's very tempting to look, for example, at this. And that means like it's very different, but it's actually the same p-value as when comparing standard deviations or confidence intervals like that. And they show also how it changes with p-values. So interestingly, so you see that confidence intervals can tolerate quite a bit of overlap with relatively small p-values. So this is something that people underappreciate sometimes. Okay, so this was my hypothesis testing, and it's 12. And I probably have another 15 minutes, so I'm not quite sure what's the best. Should we take a break? Are you ready for a break, or would you hold on for 15 minutes? Can I have a show of hands? Who wants to go?
Olga, lunch is next, so why don't we, lunch is next,
Yes.
so why don't we, if it's okay, we'll just go another 15 minutes and then we'll come back at lunch. That works.
Well, doesn't help me for the next part. Okay, let me go for 10 minutes. And we will have another show of hands in 10 minutes, okay? And see how we go. Because I don't want to, food is important. So what I would like to do now, and this is a really important part, I want to make sure we talk about this, is now that I talked to you about how great p-values are, I need to tell you how bad p-values are. And I really don't want you to go away without that, because otherwise it can be kind of misinterpreted. P-values are overused and abused, and have some controversy associated with them, to the point that last year American Statistical Association issued a statement on the appropriate use of p-values. American Statistical Association never issues any methodological statements, ever. So it was the first time when they felt really strongly, and it was actually not very easy. It was a group of experts which kind of worked together, who came from different perspectives and debated what is the right use of that. So they came up with these 6 points. So p-values can indicate how incompatible the data are with the model. Fair enough, right? Our model was that there is no difference between the groups. So if we have signal to noise very extreme, it means that it's incompatible with our assumption, right? That's good. P-values do not measure the probability that the hypothesis is true, right? Small p-value doesn't mean that it's unlikely that the null hypothesis is true. It's a different thing, right? Scientific conclusions and business policy decisions should not be based only on whether a p-value passes a specific threshold. I would add publication decisions should not be based on p-values passing a specific threshold. Proper inference requires full reporting and transparency. I'm quite sure that the paper comparing Asians and Europeans had appropriate t-test procedure, so I don't think there was anything wrong with their testing. It's more that their data had problems, right? And if they didn't report it fully, it wouldn't be possible to find. A p-value or statistical significance does not measure the importance of a result. And this is really important to know. So statistical significance is not the same as practical significance. So essentially you can have any sample, like with an arbitrary large sample size, you can make any fold changes statistically significant. Now, is it useful to you? It's not necessarily useful. So practical significance and common sense should play a very important role in addition to the statistical considerations. And by itself, a p-value does not provide a good measure of evidence regarding a model or a hypothesis. So I do see people saying sometimes, I have a small p-value, therefore my approach is correct. It's not, right? Okay, so what can go wrong with that? And let me just at least start on that a little bit. First of all, p-value is a result of our summaries of the data, right? So we build the signal-to-noise ratio, right, and we use this to infer this with respect to this reference distribution. But because our data are random, our data are subject to variation, it means that the p-values are also random. It means that if you have a small p-value today, it doesn't mean that you will also have a small p-value tomorrow with the next data set. And this happens a lot. So this is another example from another Nature Methods paper. They have two populations, just simulated data, where the difference between the groups is fairly small compared to the variation. So what they did, they took 10 replicates from one group and 10 replicates from another group, did the t-test just as we discussed, found very small p-value. Okay, fine. Now, did it again. Took another 10 from one group and 10 from another group, did it again, had a very different p-value. See, the difference goes in the opposite direction. P-value is 0.8. Same problem, right? Simulation 3, again, different fold change, p-value again very large, and so on. So depending how, every instance, depending on how the data fall out, you can have very different conclusions from the same problem. So if you have a small p-value today, don't expect it necessarily to be the same tomorrow, especially if the overlap between the groups is substantial. Now, they took it a little bit to a larger scale. Instead of doing it 4 times, they did it 1,000 times. And this is when they had sample size of 10. They plot log p-values just so that we can focus on the small values. And you see that only 186 instances out of 1,000 had small p-values, right? Now here they increased the sample size to 30, so a few more p-values are in the small range, but less than half. They increased to 64, okay, now more, and only with 100 replicates, they most of the simulations were in the range of the small p-values. So clearly sample size has a very strong impact on how reproducible your results will be. But this is another thing, and this is my favorite conclusion from their paper. They used the same experiment, but they plotted differently. So now here, the x-axis is the log p-value across different number of replicates. So as before, we see that as sample size increases, the p-values move more to the smaller range, right? So more consistently small, and the hypothesis test rejects the null hypothesis. But the y-axis now, what they do, they plot the histogram of log fold changes, right? The differences between the two groups, but only among instances where the null hypothesis was rejected. So again, what they do is they collect data, test if there's a difference between the two groups. If they do not reject the null hypothesis, they do nothing. If the null hypothesis is rejected, they claim significant difference. They keep the fold change, right? The difference between the two. But just to see what's going on, right? So now look at this. 10 replicates. They only plot fold changes, log fold changes, among things which survive the p-value cutoff. The true difference is 0.5. Look at that. There is not a single instance which is even close to the true value. So when sample size is small and they look at this fold change cutoff, either you don't find the difference, or if you do find the difference, you dramatically exaggerate the extent of regulation. Now sample size increases, now it kind of starts edging towards the true value. Sample size increases more, and only with 100 replicates, now your fold changes kind of fluctuate around the true value between the groups. So this is the problem also with underpowered studies. Not only you are not likely to find the difference, but if you do find it, you are likely completely off. So essentially it inflates your expectations as far as the extent of regulation. So we have to be very careful with that. And the same thing with confidence intervals. So now they try to do the confidence intervals, and you see that when sample size is small, it also fluctuates all over the place, and it's only when you have large enough sample size, you have consistent enough and narrow enough confidence intervals. Okay, so this is one thing. So message one is that p-values are random, just like our data are random, and therefore they will have some extent of variation. And if the study doesn't have enough replicates, we will expect to see more variability than if the study has more replicates, right? But this is one aspect of this. And now another aspect, also going back to your question, DDA versus SRM versus maybe DIA, is the number of proteins we look at. Because everything I spent like an hour now talking about, I always talked about one protein, right? But we don't have one protein in our experiments, we have more proteins. And depending on which type of workflow, the numbers vary quite a bit. Remember I was saying if we look hard enough, we find something. It's exactly the same situation, right? Even if nothing is regulated, but we test enough proteins, remember we still have 5% false positive rate, right? So if we test 10,000 proteins, we will find some false positives, right? And so this is also from the blog of the same statistician who said, well, this was a poster by somebody who did fMRI experiments on dead fish and found many active brain regions in dead fish, right? Why? Well, because fMRI is extremely sensitive, right? So they have a lot of pixels which they test for activity, and if you test enough pixels, you will always have some things which appear to be different, between, active and inactive states. So what we also need to do, we need to make sure that we account for the fact that we test this hypothesis over and over and over for different proteins. And if we want to prevent false discoveries, we need to adjust for the multiple testing, right, for the multiplicity of testing. And we adjust this by essentially being more conservative and not making decisions as eagerly as if we only have one protein. So, I think I'll have to stop here. What I'm planning to do next is to tell you about, just finish that part, right? So tell you about multiple testing and a couple other important things. And then after that, we will shift to topics which are more specific to mass spectrometric workflows. And I would like to share with you the work we do in my group in terms of statistical models and analysis workflow for targeted, but also for DDA and DIE workflows.